Picture this. It is 1347. A ship arrives in the port of Messina, Sicily. The sailors on board are dead or dying. Black swellings the size of eggs have erupted from their armpits and groins. Blood is seeping from orifices it has no business seeping from. The harbor masters take one look at the ship and order it out of port immediately. The ship leaves, the plague doesn't. Within 4 years, somewhere between 30 and 60% of Europe's entire human population is dead. Entire villages erased, cities reduced to open mass graves. A continent that took 1,000 years to build its population loses half of it in less time than it takes a child to finish primary school. Here's where it gets strange. Not everyone died. Some people walked through the middle of the worst biological catastrophe in recorded human history and felt nothing. No fever, no swellings, no blood, nothing. They watched their neighbors die. They buried their families. They carried bodies through streets that smelled of something you do not want to imagine, and their own bodies simply refused to participate in the dying. For 700 years, historians assumed these survivors were just lucky. They were not lucky. They had something written into their DNA that the plague could not defeat, and the story of what that something is, where it came from, and what it is still doing inside human bodies today is one of the most extraordinary biological mysteries in the entire history of our species. Part 1. The village that should not have survived. The year is 1665. You are in the village of Eyam in Derbyshire, England. 300 years after the first Black Death outbreak, plague has returned to Britain. The village of Eyam makes a decision that history will remember forever. The villagers voluntarily quarantine themselves. Nobody leaves, nobody enters. They do it to protect the surrounding communities from infection. They know they are probably signing their own death warrants. Of course they do, and they do it anyway. Because humans, even in 1665, were occasionally capable of extraordinary collective courage. The plague moves through Eyam for 14 months. 260 of the village's roughly 350 inhabitants die, but roughly 90 people survive. Some of them survive despite nursing infected family members directly, despite sleeping in the same beds, despite having no protective equipment, no understanding of germ theory, no medicine of any kind that would have made any difference. A historian studying Eyam centuries later notices something about the survivors. Their descendants are disproportionately represented in the modern population of that region of England. And when genetic researchers in the 1990s begin mapping a specific mutation in the human genome, they keep finding it concentrated in exactly the populations descended from Black Death survivors. The mutation has a name, CCR5 delta 32. And the story of how scientists found it, dismissed it, and then were forced to confront what it actually means is one of the most dramatic reversals in modern genetic research. But the mutation itself is only the beginning. What it does inside the human body is something nobody predicted. Part 2. The door the plague couldn't open. The year is 1996. You are in a virology laboratory at the Aaron Diamond AIDS Research Center in New York City. A researcher named Bill Farr has just identified something in the blood samples of a specific group of patients who have been repeatedly exposed to HIV, patients who by every statistical measure should be infected. They are not infected. Farr's colleague Nathaniel Landau runs the analysis again. Same result. The HIV virus enters human cells through a specific protein receptor on the surface of white blood cells. The receptor is called CCR5. It functions essentially as a doorway. The virus docks with this receptor and uses it to enter the cell and begin replication. In these patients, the doorway is broken, not damaged, not partially blocked, structurally absent. A 32 base pair deletion in the gene that produces the CCR5 receptor means the receptor never forms correctly. The doorway that HIV needs to enter the cell does not exist. The virus arrives, looks for the door, cannot find it, and moves on. Landau and Farr published their findings. The mutation is designated CCR5 delta 32. People who inherit 2 copies of it, one from each parent, appear to be almost completely resistant to HIV infection. People who inherit 1 copy show significantly slower disease progression if infected. Here's where it gets strange. CCR5 delta 32 is not evenly distributed across human populations. It is almost entirely absent in populations from sub-Saharan Africa, East Asia, and the Americas. It is most heavily concentrated in Northern and Western European populations, specifically in exactly the regions most devastated by the Black Death between 1347 and 1353. A geneticist named Stephen O'Brien at the National Cancer Institute does the math. The mutation is too common in European populations to be explained by random genetic drift alone. Something selected for it. Something killed the people who didn't have it at a catastrophic rate and left the people who did have it disproportionately alive to reproduce. The Black Death didn't just kill people, it edited the human genome, and it left the edit in every descendant of every person who survived, which means it may be inside you right now. But the mutation itself raises a question that neither Landau nor O'Brien could fully answer. If CCR5 is a doorway that HIV uses to enter human cells, what was the Black Death using it for? Because Yersinia pestis, the bacterium that causes plague, is not a virus. It does not infect cells the same way HIV does. So why would blocking a viral entry point protect against a bacterial infection? The answer involves a mechanism that researchers are still debating today, and it goes somewhere nobody expected. Part 3. The immune system's ancient architecture. The year is 2008. You are in a genetics laboratory at the University of Liverpool in England. A research team led by Christopher Duncan and Susan Scott has spent a decade arguing something that the mainstream academic community has spent a decade dismissing. Their argument is this: the Black Death was not caused by Yersinia pestis alone, or perhaps not primarily. The speed of transmission, the mortality rates, the patterns of spread documented in historical records from the 14th century do not match what modern epidemiologists observe in documented Yersinia pestis outbreaks. Modern bubonic plague caused by Yersinia pestis requires flea vectors to transmit between humans. It spreads relatively slowly. It requires dense rat populations in proximity to human settlements. The Black Death spread in winter, in cold climates where rat populations were low, in Iceland, which had almost no rats, between households with no apparent contact, at speeds that modern Yersinia pestis simply does not achieve. Duncan and Scott proposed that the 14th century epidemic involved a hemorrhagic fever virus in addition to or instead of Yersinia pestis, something that spread person to person through respiratory transmission, something more like Ebola than plague in its clinical presentation. The academic community dismisses them, of course they do. Overturning the established cause of the most famous epidemic in history is not a career move most researchers are eager to make. But then something happened. In 2011, researchers extract ancient DNA from the dental pulp of 14th century Black Death victims buried in a documented plague cemetery in London. They find Yersinia pestis DNA confirmed. The bacterium was there, that part is settled. But the CCR5 delta 32 mystery remains. Because CCR5 is not a known entry point for Yersinia pestis. The mechanism by which this mutation would protect against a bacterial infection is not straightforward. And the selective pressure required to drive the mutation to its current frequency in European populations through plague alone requires mortality rates that even the Black Death's worst estimates struggle to justify. The door is confirmed. Why the plague couldn't open it is still not fully understood, and the next discovery makes the whole question significantly stranger. Part 4. The mutation older than the plague. The year is 2003. You are in a population genetics laboratory at the University of California, San Francisco. A researcher named Alison Galvani has run a mathematical model that she publishes in the journal Nature. Her conclusion is precise and deeply uncomfortable for the established narrative. The frequency of CCR5 delta 32 in modern European populations is too high to have been produced by the Black Death alone. The math doesn't work. Even assuming a mortality rate of 50% and perfect selection for the mutation, the Black Death was not long enough, was not sustained enough, and did not recur frequently enough to drive the mutation from near zero to its current frequency of approximately 10% in Northern Europeans. Something else was selecting for this mutation, something older, something that was killing people without CCR5 delta 32 at catastrophic rates long before the Black Death arrived in Sicily in 1347. Galvani runs the models for different historical epidemics, different pathogens, different timelines. She proposes that smallpox, endemic in European populations for potentially thousands of years before the Black Death, may have been the primary selective pressure. Smallpox, a virus that uses a cellular entry mechanism related to CCR5. The same doorway, different virus. Much longer timeline. Her paper is published, it is noted, it is debated, it is not resolved. Because here is the problem. If CCR5 delta 32 was already present in European populations before the Black Death because smallpox was selecting for it for centuries, then the survivors of the Black Death weren't just plague resistant, They were the product of thousands of years of epidemic selection going back to the earliest days of settled civilization. Every major epidemic in European history, smallpox, plague, possibly others not yet identified, each one killing the most vulnerable. Each one leaving the carriers of specific mutations more likely to survive, more likely to reproduce, more likely to pass the mutation forward. The human genome is not just a biological blueprint, it is a record of every catastrophe that tried to erase us and failed. And we are only just beginning to read it. But the final question is the one that nobody has answered, and it involves something sitting inside your body right now. Part 5. What the survivors left inside you. The year is 2023. You are in a genomic research facility at the Wellcome Sanger Institute in Cambridge, England. Researchers are running the largest population-level analysis of CCR5 delta 32 frequency ever conducted, mapping the mutation across modern European populations, correlating its frequency with historical plague mortality records, building the most detailed picture ever assembled of how the Black Death edited the human genome. The preliminary findings are extraordinary. The mutation is not randomly distributed even within European populations. It is concentrated in specific regions that correspond with documented high plague mortality in the 14th century, regions where the selection pressure was most intense, where the dying was most complete. The areas that lost the most people in the Black Death produced descendants with the highest frequency of CCR5 delta 32. The catastrophe and the survival are written in the same map, and this is where everything changes, because the researchers also find something they did not expect. In populations with high CCR5 delta 32 frequency, there are measurable differences not just in HIV resistance but in baseline inflammatory immune response profiles, in susceptibility to certain autoimmune conditions, in the way the immune system calibrates its response to novel pathogens. The Black Death did not just select for plague resistance, it may have subtly reshaped the entire immune architecture of the populations it passed through. The immune systems of Northern Europeans today, with their elevated rates of certain autoimmune conditions, their particular patterns of inflammatory response, their specific vulnerabilities and strengths, may be in part the product of a catastrophe that ended 670 years ago. The plague is gone. Its fingerprints are not coalescing. So why were some ancient humans completely immune to the Black Death? Part of the answer is a mutation, a 32 base pair deletion in a single gene that removed a cellular doorway and left the plague with nowhere to enter. Part of the answer is older epidemics. Thousands of years of smallpox and other pathogens slowly selecting for that mutation across generations of European populations before the Black Death ever arrived. Part of the answer is still unknown. The precise mechanism by which CCR5 delta 32 protects against Yersinia pestis is still not fully mapped. The debate over what the Black Death actually was at a virological level is still not closed. The full scope of what the epidemic did to human immune architecture is still being calculated. But here is what is settled. The survivors were not lucky. They were the product of thousands of years of biological selection. Every epidemic that came before the Black Death, every pathogen that killed those without the right mutations and left those with them standing, contributed to building the immune architecture of the person who walked through the Black Death untouched. And those people are your ancestors. You are here because someone in your lineage had something the plague could not defeat. Maybe the mutation, maybe something else we haven't identified yet, maybe a combination of factors that modern genomics is only beginning to map. The Black Death did not just kill half of Europe. It selected the other half, and that selection is written into every cell of every person alive today who descends from those survivors. The next time you feel your immune system responding, the fever, the inflammation, the precise orchestrated biological war your body launches when something foreign arrives, that system was not built in a hospital, it was built in the middle of the worst catastrophe in human history by the bodies of people who had never heard of a gene, who had no concept of a mutation, who simply had something inside them that the dying world could not take. They didn't know what it was, they passed it forward anyway. The question that remains open is not what saved them. The question that remains open is what else they survived that we haven't found in the records yet. What other plagues, what other selections, what other catastrophes are written into the genome that modern science has not yet learned to read? The door is still open, the archive is still being searched, and the answers when they come will be written in the oldest language that exists, your own biology.